So thank you for the opportunity. So as Irina was talking about, this will be more of a follow-up of what Patrick spoke about for a couple of weeks ago. So just to remind you what's happening. So we were then at the European Expo, where we were performing experiments of time-resolved solution scattering. So our setup was something like this. So we had the liquid jet coming in here, we had our pump laser, and we have the megahertz repetition rate X-rays coming in. So what we're doing is that we're collecting the data with a rapid readout from the detector. So at the European Expo, we have this weird pole structure that are comprised of small bunches. We have a gap and then small bunches again. So the idea behind it was, okay, so what if we put in pump within the train and then we start collecting so we get a time series out of this. So that's the premise of the experiment that we did. And Patrick went through then all the details. So if you were not there or so, I suggest looking at his talk that he made, where we go more into detail on how it works exactly, the setup and how it is. So let's move on to data analysis instead. So the protein we're looking at is the LOVE2. So it's a light oxygen sensing voltage domain from the Avena sativa, so oat. So it is a blue light receptor with a FMN chromophore. So we have the huge J alpha here that is usually connected to a serine threonine kinase in the phototropin, which is a larger protein. So the idea behind this is that we have blue light shining on the protein, and then we have the photoactivation. And 2 milliseconds after that, we have a cis adduct formation. So we have a covalent bond between the cysteine and the FMN. And then some microseconds after that, we have some sort of unfolding the attachment, which was first seen in NMR, but then there is no direct structural evidence for this in using X-ray techniques, for example, because you cannot see it in the crystal because of the crystal packing. But through spectroscopy, it's been seen that something happens on the time scale that hopefully we could record them. So for the data collection, we were collecting from interchangeably one light, one dark, one light, one dark. We did this because we wanted to keep any possible systematic errors within the train to keep it consistently throughout. So if something happened so for example, if we have a certain shot noise at the fifth pulse, we know that is consistent always in the fifth pulse, then we will have it in the dark and both in the light. So we do the normal standard way of the analysis, the data of getting the 1D curves. So we do radial integrations, and then we have the data curation where we have rejecting outliers. For example, if the jet is unstable sometimes, then we need to reject those, and then we have the normalizations done. And then after that, we separate our data into dark and light, and then we calculate the different scattering for them. So we do this for each of the time points that we have. In our case, we have 175 of them. So when we look at the data, what we had in the data, we can see, okay, we see the evolution throughout the time. So we have the blue lines, which is the negative time points that we use as a control to see that we have no excitation. And then we see the evolution of the signal as further we go into the time. As you can see, we have a huge signal here at the top. So it's just been scaled down here for visualization. So you can see that this is the dominant feature, this first peak here. And as indicated by the axis, we have very low noise levels actually for this experiment. So then if we want to interpret this, how do we do it? Well, so the idea was, okay, we can do the same way as for the normal data. We can have a set of models, which we can calculate the scattering of, and then we can compare this with experimental data. So in this case, we chose to use PepsiSax, and that's because it was conveniently fast for doing a bunch of computations at the time for the models we had. But the biggest problem here was the generation of the models, because we know the dark state from the crystal, and from just a normal AlphaFold prediction, but we do not know the light state of this, since there is no other structural models available for this. So first we tried out AlphaFold with aggressive sampling. So aggressive sampling in this case means that the dropout rate of the training has been put on, which enables basically the network to put out different kinds of structures that are basically more different than the end structure. So when we tried this approach, we could see, okay, we actually don't see any big difference. So I actually tried to fit this at first, but we could actually see that there is no good fit because it's such small movements we're looking at, and we actually have larger ones. So instead, I had the idea, okay, what if we can somehow manually make the helix unfold? So then my idea was, okay, what if I insert some mutations throughout the helix? So I had then 3, 5, 7, 9, 11 starting from the bottom to the top. And the idea was, okay, what if I insert glycines here just to see if I could destabilize it, for example. So I did then the same procedure again with the AlphaFold with the aggressive sampling plus the mutations then as my input sequence. And then I could generate something that looks like this. So then we got, okay, we can see that the helix is getting unfolded this time. So after this was done, I just mutated the residues back from the AlphaFold just to get the same sequence as the starting one, as the original one. So now that we had the models, okay, so now we can proceed with the whole workflow of calculating the scattering and then comparing it. So the next important thing here was the comparison to the experimental data. So what are we actually observing in different scattering? Well, the different scattering contains information of the magnitude of the change that we're looking at. So for example, if we would have something that is not changing at all, so for example, if we have here to the left, which is just dark minus dark, if we have something that J alpha is completely intact throughout, we will have low or no signal. And if we have somewhat that is, okay, we have some sort of half unfolding, we might see some signal. And then if we have a huge unfolding, we would have a greater signal. And this would also be a shift in the Q range, which might be difficult to say what is what because they are so close to each other. Another thing that affects the signal is actually the number of molecules that are excited. So if we have no molecules excited, we will have the same as before, almost like dark minus dark, whereas there is no signal. But if we have some molecules that are excited and then we will have some sort of signal. And then if we have a larger number of molecules, we will have even more. So it works as a scaling factor that increases the signal. So now the important part was, okay, we need to take this into account when we want to do our modeling. Because we want to model our movement that we're watching accurately. So we want to know, okay, are we seeing, okay, you saw a mistake here, but we should be basically, the idea is that we have many molecules and a short movement of the helix, just semi-unfolded, or we'll have the vice versa. We will have a huge movement with few molecules excited. As you can see, this should be swapped around. And this could then give the same signal in theory. So we want to be able to discriminate this and find what is much more, which one it should be that is represented in the sample we're looking at. So in order to do this, we use steady state SACs to determine the photo excitation yield. So we went to Diamond and performed an experiment where we just had steady state SACs where we were eliminating our samples for different time periods. So we could see that we had saturated the sample to basically 100% excitation yield. So you can see here the dip here is saturating almost after 10 seconds. So we were able to see that this is happening because we have it, as I told you, FMN. So here we see the absorption spectrum, so the UV spectra. And we know that when we have the sample illuminated, we can see the peak going down. So we know that, so we can confirm, so we know that we can reach up to 100% from the spectroscopy. So once we had our sample, so once we had the 100% sample and we had a SAX curve, steady state SAX, we wanted to carry this over to our time resolve data. So what we had to do first here is that we had to scale the data. So here you see in blue, we have the absolute SAX curve that we measured from the synchrotron at diamond. And then here we have the green one here is the XVL data. So this is just the dark, and then we apply the scaling factor to put them on the same level. So we have both, so we can make it comparable. So then we did the same with the difference. We apply the same scale we have here to the difference data. So here we have the difference data from SAX, the 100%. And then we have here for our latest time point from the time resolve data. So when we apply the scaling factor, so then we would have a difference between them, and then we can apply a second scaling factor, which should correspond to the photoexcitation yield. And in our case, we estimated to be around 15% the activation yield that we were able to get at the European XFL. So now that we know, okay, now we know the photoactivation yield, we can try to use this to get some structural information. So we had the idea of doing exactly the same as before, but this time using the computed scattering. So we have the computed scattering is on a scale, it has units. So we wanted then to put the experimental data on the same units as before. So then we have the computed scattering, so the blue one, and then we have the unscaled experimental scattering from the XFL, and then we apply the same thing. We get a scaling factor that we can apply, and then we will apply the same scaling factor onto the difference data. So here we have a computed difference scattering curve between two models, and here we have the experimental data here. So when we have applied the first scale, we see here, okay, the second scale that we need to apply, which should correspond to a so-called refined yield or projected yield, which should be something that we can get. And this yield then we should be able to use this yield to compare to our actual yield to find good or bad models in our data. So, okay, so if we then compare our data, we want to use two metrics in our case. So we have one, which is the photoactivation yield here on the y-axis, and then we have the R-square here on the x-axis. The R-square is just a measurement of how similar the two curves are. So basically just the sum of the residuals, more or less. The square sum of the residuals. So from this, we could see, okay, we want to maximize the R-square for the model, but we also want to find something that is within reason of the photoactivation yield that we determined or estimated that we had at the XFL. So anything above activation yield of 1 would be nonsense, because that's not feasible at all. So you can see that we have quite a lot of models that are that had a photoactivation yield of 1 or above. And we're actually looking into our data, we can see that we all have a small cluster here forming, which is very close to the determined photoactivation yield we had at the European XFL. So if we then look at the curves, how they actually look, we can see that are actually quite similar to the experimental state here. So we have the experimental data here in blue, and then we have in the shades of gray the best models. So everything that's in this small box here, it corresponds to plus minus 5% of the excitation yield. So now that we had our best models, we want to see, okay, how do they look like? What is actually happening to them compared to the crystal structure? So here we have, so this is how the crystal structure look like, which has a rate of duration of around 15 angstrom. And when we compare the difference of radius of duration between the light states here and the dark, we see that we actually get an increase, which corresponds to the unfolding of the helix. So what I'm showing you here are the models that are here in the box. So these are around 6,000 models out of 20,000. So these are what you're seeing here. So these are the best fits that we could get. And these are the RGs of the RG increase of them. So you can see that, okay, at around 6 angstrom 5, 6 angstrom, we get a plateau where we reach, okay, this is how much the helix is unfolding. So it's reached out to the maximum that we can see. We also did a similar, in addition to the J alpha that we had unfolded, we also looked at the N terminus, but we could not, unfortunately, see any difference if this was unfolded or not, because this is also something that's been debated within the field to see if this has something to do with the J alpha unfolding, but unfortunately our data couldn't really discriminate between this or not. So we had a combination of both the J alpha and the N-terminal helix unfolded. So what we actually got out of this experiment in the end is, well, we had first of all the development and establishing how to perform this type of experiments at European Expo, to use this megahertz repetition rate to get a time series. We did some modeling using AlphaFold and also incorporating the photoactivation yield into our modeling to get even more accurate models to represent what's happening in the solution. And then something I didn't talk about, but that Patrick mentioned was that we talked about the photocycle of the LoV2 domain and how we actually found out the intermediate state and could see that after 300 microseconds, the helix is almost unfolded. And that's more or less for me. So this was a huge effort for a lot of people. So I have special thanks to the people that we work mostly with post the beam time, which is Sebastian, Felipe, Patrick, and Alfredo. And then, as you can see, this was a huge collaboration of a lot of people working at the beam time, both beam time scientists and other people joining with their expertise to make sure everything is running, to make sure we had the correct parameters for the jet and for the actual nozzles that we use. And so a special thanks to them and also to the European Expo and to Diamond, where we perform experiments. And thank you all for listening.